Every day, thousands of animals are used for testing in the medical field. But is it really necessary? Animal testing has been used in the medical field for centuries, with the earliest recorded experiments dating back to ancient Greece. Today, animal testing is still a common practice in medical research, with millions of animals used each year for testing and experimentation. While many people believe that animal testing is necessary for medical advancements, others argue that the practice is cruel and unnecessary, and that there are alternative methods that could be used instead. Let us explore both sides of the argument and try to answer the question, is animal testing necessary for medical advancements? Animal testing has been a critical tool in medical research, providing invaluable insights into the human body and its response to various diseases and treatments. Proponents argue that animal testing is necessary because animals have a similar biology and physiology to humans, making them valuable models for studying the effects of drugs and treatments on human organs and tissues. Animal testing has been used extensively in the development of life-saving medications such as insulin, antibiotics, and chemotherapy drugs. For example, insulin, a hormone that regulates blood sugar levels, was first isolated and purified from the pancreas of dogs in the 1920s. This discovery led to the development of insulin therapy for people with diabetes, saving countless lives. Antibiotics, such as penicillin, were discovered in the 20th century through testing on mice, paving the way for the treatment of bacterial infections. Animal testing has also been used in the development of vaccines for diseases such as polio, measles, and rabies. Vaccines work by stimulating the immune system to produce antibodies against a specific disease. Animal testing has played a critical role in the development and testing of vaccines, helping to ensure their safety and effectiveness before they are used in humans. In addition to medications and vaccines, animal testing has also been used in the development of medical devices such as pacemakers and artificial joints. These devices are designed to replace or support damaged organs or tissues in the body. Animal testing has helped to ensure the safety and effectiveness of these devices, allowing them to be used in humans with confidence. Animal testing also allows researchers to study the effects of drugs and treatments over a long period of time, which is not possible with human subjects. This is particularly important in the development of cancer treatments, where long-term studies are necessary to evaluate the safety and effectiveness of new drugs. Animal testing also allows researchers to study the progression of diseases and to understand the underlying mechanisms that cause them, which is critical for the development of new treatments. Opponents of animal testing argue that the practice is not only cruel, but also unnecessary. The animals used in testing are often subjected to painful procedures such as injections, surgeries, and toxicological experiments. These procedures can cause physical pain and emotional distress, and animals are often kept in small cages for extended periods of time, which can lead to boredom, stress, and depression. Many animal welfare organizations and activists argue that the pain and suffering caused to animals during testing cannot be justified by the potential benefits of the research. They argue that animals have a right to live without being subjected to unnecessary harm, and that there are alternative methods that could be used instead. Some of these alternative methods include in vitro testing, testing on human cells grown in a laboratory, computer modeling, and human clinical trials. There are also concerns that animal testing may not be reliable. Animals are not identical to humans, and there are many differences in their biology and physiology that can affect the results of experiments. For example, some drugs that are effective in animals may not work as well in humans, and vice versa. Additionally, the stress and pain caused by testing can alter the results of experiments, making them less accurate. Opponents of animal testing also argue that many drugs and treatments that have been developed through animal testing have been found to be ineffective or even harmful to humans. For example, thalidomide, a drug that was tested on animals in the 1950s and 1960s, was found to cause severe birth defects in humans. It is argued that alternative testing methods, such as in vitro testing, may be more accurate and reliable than animal testing. In vitro testing is a method of testing drugs and treatments on cells in a laboratory setting. This method involves growing human cells in a petri dish or other type of container, and then exposing them to the drug or treatment being tested. This method allows researchers to test the effects of the drug on specific types of cells, such as cancer cells, without the need for animal testing. In vitro testing can also be used to test the toxicity of drugs and treatments, as well as to study the mechanisms of disease. Computer modeling is another alternative to animal testing that is becoming increasingly popular in medical research. This method involves using computer models to simulate the effects of drugs and treatments on the human body. The models are based on data collected from human trials and other sources, and can be used to predict the effects of drugs and treatments on different populations, such as children or pregnant women. Computer modeling is particularly useful for testing the effects of drugs and treatments that may be too dangerous or unethical to test on humans or animals. Human tissue testing involves using tissue samples from human volunteers to test the effects of drugs and treatments. This method allows researchers to study the effects of drugs and treatments on human tissue in a laboratory setting. The tissue samples can be taken from a variety of sources, such as blood, skin, or organ tissue. Human tissue testing can be particularly useful for studying diseases that are difficult to replicate in animals, such as certain types of cancer. Other alternatives to animal testing include microdosing, which involves testing very small doses of drugs on humans, and the use of human volunteers in clinical trials. These methods are becoming increasingly popular in medical research, as they offer a more ethical and reliable alternative to animal testing. The use of animals in medical research is a controversial topic, with arguments both for and against its use. While animal testing has played a crucial role in the development of many life-saving medical treatments and technologies, there are concerns about the cruelty of the practice and the reliability of the results. Alternatives to animal testing exist, but there is still much research to be done to determine their effectiveness. Ultimately, the question of whether animal testing is necessary